This is a tutorial for the Bioinformatics Resource Center RNAseq analysis tool. So this is a tool that allows you to interact with your data that's coming right off of our pipelines. Again, this is RNAseq, and so any sample that comes off our pipeline that's attributed to you should show up in this table below. So in addition to the table of samples, there's a number of other sort of options to interact with the samples, as well as these tabs here on the left-hand side. That should now all be blank, except for the report tab, which we'll get into a little bit later. But a lot of these tabs just require you to load some samples before you can actually do anything. Another important feature of this app and page is this report issue button, which you can hit, enter a message, hit submit, and this will send us a message directly. Typically, this will be useful if something does go wrong with the app, you'll get a message that says something has gone wrong. Then you can still hit this button, and this will send us an error report, which will allow us to try and diagnose what the problem was and prevent it from happening in the future. If you do hit submit here, it does restart the app, so this is probably something you shouldn't do if you're in the middle of analyzing some samples. And so again, this table down here is kind of the important part of this page that should have all of the samples that you've run through our pipeline. If there are samples you think that should be there that aren't, just reach out to us and we can take care of that. So this table, you can adjust how many entries you see on the front page. This is searchable, and what it contains is the sample name, which essentially each of these is a FASTQ file, the group that sample is part of, and so this is a group that was added before the pipeline run. So far there's only one group, and one thing that we'll get to in a little bit that should be useful with this app is you can add different groupings and change grouping names and stuff like that. The project is essentially tied to each pipeline run, so another useful part of this app is hopefully trying to compare samples across different pipeline runs. The data is added to our database and the species for that pipeline run. For this app, you can't choose two different species, so make sure, and it'll give you a warning, but make sure you do choose samples that are all the same species. And so just going through the boxes of this page, this first box is a project name, so every time you load new samples in the app, all of the reports and information and QC data that's tied to those samples will be listed in our database under a certain project name, and that's how you can retrieve them later on. So you can automatically, it's your name and your lab name, but you can change that for every project if you want. In order to do the differential expression analysis, you need to select samples. You can do that by clicking individually. You can hit Shift and highlight all the rows, as many rows as you want, and then hit the Select Highlighted Samples and Groups button here, and this will give you a summary of your groups and how many samples are in each group. One thing about this particular set of samples that we know we want to change is this TAM group. This is a treatment that we know induces senescence, and senescence is a much more intuitive name, so we're going to go through changing the name of that group. If you ever want to deselect a large number of, or any of the rows in this table, you can just hit Clear Selected Samples. So to change the groups, you can do one of two things. You can either just click directly on that cell and change the name. The name is changed, or you can use this grouping drop-down. And this second option here allows you to change the name in the column, so you just highlight the rows for which you want to change the name, select the group, Here we only have one group, so there's only one option, but if you have more groups, you'll have to select the group, and then whatever you want to call, or whichever group you want to put those samples into. So do that, and you can see these have changed. Another thing you can do in this set of options is to add a new column. And you just add the name of your group here, click this button, it will add a blank column, and then you can add groups in the same way I just showed you before. Let's, for example, here maybe you want to, instead of having these two shRNAs as separate groups, you want to put them all as one. So we'll highlight all those rows. This new group should now show up in this drop-down, so we'll select it, give it a name, and the new group name should show up there. So just we'll carry over these names here. And so these last two options down here are also potentially useful. They allow you to save one of these grouping columns for future use. Maybe you go through a lot of samples and alter the grouping names or add groups, and you either don't want to go through that next time, you just simply want to remember what they are. You can save a group. So the saving button, the saving group option is this last drop down here. So we're going to save this new group, click Save button. It'll tell you that it's happened and tell you the name that it's saved under. And it's simply just the name of the column with a timestamp. Once it's saved, it should show up in this drop down here. So it's this new group column here with this timestamp. And so if you ever wanted to add an old column, previously saved column to your current table, you just click on it, hit this button, and it should show up in the table here. It's just the same as this little one we obviously saved it under, but it should have this new name with the timestamp on it. Okay, so now that we have our grouping set, we're going to move forward and do differential expression, but still using the original groups with the new name for the senescence group. And so we're going to use all the samples, so we'll select all the samples. To do differential expression, you have to choose the group column that you want to use. We're just going to use the sample group column, hit the select highlighted samples and groups button, and this will, again, as we saw before, show you the summary with the number of samples per group. So this all looks good. Once you're ready to do the differential expression or start the analysis, you can click this button here. You can also just do quality control. This is generally a little bit quicker, but with this button you'll get differential expression analysis and quality control, so we're going to do that. And so what this is doing right now is it's using a software package called DESeq2, which is actually doing a lot of the statistics, dispersion analysis, and fitting the model for your groups to allow us to, in the future, do direct comparisons between those groups. So now our samples have been loaded and we're ready to start the differential expression comparison between groups. And you can see the samples that we selected are shown again in this table here. And we'll extend the number of entries so we can see all 12. The project and also the sample group that we selected from the previous tab. A couple other things that happen behind the scenes as the samples are being loaded. Can be seen in this file download IGV links tab, where the samples that you chose are listed, and this is where you can download the counts file, or perhaps more importantly the bigwig file for each sample that you chose. And you can also click this button to view each sample on IGV. IGV should be open, or needs to be open, for this link to work. If IGV is open, it should go right to IGV, and you should be able to see the bigwig track. If it's not open, I would not press this second button because it will just refresh the app, and you will lose your place. Further, there's some quality control metrics where each quality control for each sample is done side by side, which can really point out or make obvious whether one sample sort of looks out of place, which can be hard when you're looking at each sample individually. And a lot of these figures you can hover over and get some more information. And there's some FASTQ quality control and some base quality. And here's an example where you can kind of see, things start to stick out for certain samples, and it becomes pretty obvious when they're all next to each other. And lastly, there's some alignment metrics as well. And so moving on to differential expression, as we said, these are the samples you've loaded. There's also a PCA plot where you can hover over the points here and see the individual samples. And they're originally colored by group, but you can also map on some of the quality control metrics that we saw in the previous tab and see whether something like duplication rate could potentially explain some of your variance as opposed to some sort of biological effect. Here it looks like the color is kind of spread across groups or within groups, and so it looks like at least duplication rate doesn't seem to be explaining the variance that we see. So we'll go back to group. This is a distance matrix for the expression of all genes, clustering the samples and seeing whether any particular samples or groups of samples sticks out. So to then actually do a differential expression comparison, you choose the groups over here. We'll choose wild type as our baseline and senescence as our test group, and click this button. And the software DSEQ will work behind the scenes to identify any differentially expressed genes. And so initially, this is run with the p-adjusted value cutoff of less than 0.05 and no log 2-fold change cutoffs, and the number of genes are shown up here. The PCA plot and distance matrix are still here as well as the table of samples, and they can just be expanded by clicking the plus sign there. You can change the cutoffs here, so we'll do a cutoff of 2-fold or log-fold change of 1, both negative and positive, and you can re-render the heat map, and you'll see as expected the number of differential expressed genes go down. Your heat map. The heat map is of only the differentially expressed genes. All of the expressed genes, along with the differential statistics, are shown in the table here. This table can be searched. You can also search the individual columns for symbols or IDs. And there's also an MA plot and a volcano plot here with the cutoffs that you had picked above, significant genes colored in red. There's a few things you can do to modify this heat map to get a little more information and also to affect where you go later on in the app. You can click this button up here and some options appear where you can remove and add back samples to the heat map, and it'll re-render the heat map without those samples. You can also map on other grouping columns that you added in the first tab to your heat map, and this will add an annotation row for the columns for that particular grouping. And probably more interestingly and importantly for everyone is the ability to cluster your heat map. And here we'll change this to 2 clusters. This is hierarchical clustering, and you can re-render this heat map with these new parameters. we'll hide the window with that button. You can see we have our additional annotation bar in 2 different clusters, where here we're looking at these senescence samples versus the wild type samples, and you can see we have 1 cluster that goes up in the senescence and 1 cluster that goes down. And additionally, you'll see a column is added to the table with the clusters, and so you can filter by cluster if you like. And also tabs have been added to both the MA plots and the volcano plots, coloring the particular clusters. Here it's pretty obvious. There's 1 that goes down and 1 that goes up. But with more clusters, you can kind of pull out what each group looks like visually here. Another thing that you can do in this table here is to highlight particular genes. You can search for genes that you're interested in and highlight them like so, and then click this button, and it'll highlight these genes in all 3 of these heat plots, all expressed in each cluster. Additionally, you can clear the selections here. Additionally, you can either enter or paste a list of genes into this box here. For example, let's just use some genes that are in the table above, and these can be comma separated or space separated. If you click this button, you'll both get a new table with just these genes to get the differential statistics, and you'll also get a separate tab in this plot below where you actually see where these genes fall. And all of these figures and tables will be included in any reports that you finish at the end or generate at the end, and we'll go over this in a little bit. Additionally, with this box here, this is probably more useful, not necessarily for identifying 2 genes, but you can get a gene list from some other source that's either comma or space separated and just paste it directly into here and get a separate table for just your genes of interest, whether it's a gene ontology category or some differentially expressed gene list from another paper. And you can visualize those genes below. So now that we have our differentially expressed gene set, genes that go up or down by log fold change 1 or more than -1 in the other direction, we can move on to functional analysis. So now we've moved to the functional analysis tab, where we can see the same heat map that was rendered in the previous tab re-rendered here. We have all of our differentially expressed genes and 2 different clusters, one with genes that are going up in the senescence group and one with genes that are going down. And so there's a few functional analysis options on this page. One being kind of a Fisher test-like overrepresentation analysis, which basically is just going to look at whether there's a statistically significant overlap between your gene list and the gene list that you're comparing it against, or gene set enrichment analysis, which is going to look at the entire rank list of your genes and see whether the gene list you're interested in comparing it to is more enriched in the top, the genes that go up or the genes that go down. You can see that if you do clustering, the clusters carry over here, so you can do the overrepresentation analysis not just for the entire gene set, but each individual cluster. If you look at this drop down, there's a number of options for you to choose from. This custom gene list from database is going to look at all the gene lists that you've created through previous runs in the app, and that's going to get genes that go up or down for each differential comparison, in addition to any clusters that you might have made. There's also the traditional gene ontology categories, KEGG, and also the entire list of MSIG-DB gene sets from Broad, which include things like pathways and cell types and stuff like that. So these can easily be chosen from. We'll do the more general hallmark gene sets from msigdb. And this is a pretty small gene set, so it'll be quick. Other gene sets, such as the gene ontology categories, can take a little bit of time. So once you run your analysis, you get a table here with tabs for either the entire gene set or each individual cluster. And you get a description of the group and the statistics about that comparison with p-values, in addition to the genes that are actually responsible for that overlap and for that enrichment. And as I said, you get separate tables for cluster, in this case, cluster 1 and cluster 2, so you can see, here that it's genes that go up or down, so you can really tell whether there's a difference in which, whether the up or down genes are contributing to the enrichment in your entire gene set category. It also automatically creates this network plot, which is basically looking at whether there's certain groups that have overlapping sets of genes in them, or certain gene lists with overlapping sets of genes. And sometimes this isn't as useful as in here. You can tell there's not a lot of overlapping genes, so there's not a lot of connections between groups. But things like gene ontology, which we can do right now, are built as such that there's lots of overlap and lots of relationship between groups. And this can be particularly helpful because for certain types of analyses, you get a lot of terms that seem very similar, and these network plots can help you identify large groups of terms instead of individual terms themselves. So now the gene ontology analysis is finished. We get a similar table as we did for the hallmark gene set. And we get lists for both the entire set of differentially expressed genes in addition to each individual cluster. And one thing that stands out is that you get, with the gene ontology, since there's a lot more relationships between the categories, nice sort of defined clusters of terms such as angiogenesis, cell cycle, and DNA replication. So this, in combination with the list, is particularly helpful because the list, as you can tell, has sort of a lot of very either broad terms that don't mean too much, or lots of redundant terms. So as we said at the outside of this page, we can also do gene set enrichment analysis. And we'll use the hallmark gene set, which again is pretty small and pretty fast. And so we get a similar table, except we have some slightly different statistics, such as enrichment scores, and also the leading edge of genes that are most responsible for that particular enrichment. We get a network plot. Another feature of the gene set enrichment is these GSEA enrichment plots, which you might be familiar with. So here you can select any number of rows, hit this button, and below you should get these enrichment plots, which show you which side the enrichment falls on, and also will show you the differential expression statistics for the leading edge genes that are responsible for those enrichments. And all these tables and figures will be in the output if you make a report, both in the HTML report and as a separate PDF. And so we can move on to actually making a report in this Reports tab. So the Reports tab allows you to do two things. You can either generate a report from your current analysis, or you can access and download old reports from previous runs through the app. And you can both either so you can click on for old reports, you can click on a row and just show that report, or you can also click this button while you have a row selected and download all the files associated with that selected report, such as QC, differential expression, functional analysis, as well as the report and the figures. And so this is what generally a report looks like. It includes all of the figures that you generated as you went through the app, lots of important statistics, and also importantly, it includes any code that's required to generate these figures, as well as all of the parameters that are required for anything that's created in this report. So this allows you to potentially go through and create these yourself and modify them in certain ways. It also helps you understand how they were created. And at the bottom, we also include all the packages and their versions, which can help with methods for papers. And also if we want to go back at some point in the future and recreate what you have in this report. So here we're going to make a report from the session, the app session that we're currently in. You can change this text here to give it a more sensible name. We'll do wild type versus senescence, 2 clusters, and a timestamp will also be appended, so you don't have to worry about sort of overwriting anything. And then we click this button, and it'll start making the report. And as the report's made, you'll get these updates as to where it is. And so this is going to go back and regenerate everything, running all the code over again, which can help with reproducibility. And so it can take a little bit of time if you went through and did a lot. So for example, we went through and did that gene ontology functional analysis, which took a little bit of time. So this will take the same amount of time. But the way that this is run, you can, as the report's being made, you can go back and kind of edit and add to your analyses and do certain things within the app and look at things that are currently open, or even start a new analysis while the report's being generated. And then you can come back and get updates, and when it's done, it'll tell you it's done. The report will be shown down here, and that report will also be added to this list. And we can also download all the reports and files that are related to this particular project. So now our report is finished. It's loaded down here. And you can see all the figures that we created. Sometimes it can take some time to load. So this includes all of the plots for the different clusters, even the genes of interest, and all the different functional analyses, including the separate ones for clusters. And so another important thing is the ability to download all the reports and files. So you can click this button to download all of the files and figures and reports that are associated with this project. So if you had made multiple reports, they would all go into this download. And we'll see what it saves in a second once it actually downloads. So this is the directory that we downloaded. It has all the counts for the samples that you selected, the reference files, the RM block has all the R programs and packages that were used, some quality control summaries, the TPM table, and also all of the files and figures associated with each report. This is the report we just made. You can see there's the tables from the functional enrichment analysis for both GSEA and the overrepresentation with individual ones for the clusters. These are the results of your differential expression, both the just the differential genes and the unfiltered table. These are GMT files with your gene lists from your functional analysis. This is the actual report. And then you also have all the figures that were generated during this run through the app. So that's all we're going to show you today. If you have any questions, don't hesitate to reach out to us. And thank you for using the app.